Thank you for the invitation and the opportunity to present my work. So I'm Thomas Renne. I'm a PhD student at the University of Montreal. And today I'm going to present you scFunBurd, a new method to quantify the cellular liability for complex disorders of rare gene-disrupting variants. And I will show you how this method can be useful in autism research as an example. So first, let me briefly describe what is autism, just to know, to have the basics. So the autism spectrum disorder, or ASD, is a neurodevelopmental disorder, and it's usually noticed around the age of 3 years old, and it's characterized by two main traits: some difficulties in social interactions and stereotype behavior with restricted interest. And the thing is, ASD is also highly linked with other comorbidities like epilepsy or intellectual disability, making it difficult to study due to these different interactions. And on the genetic, and also ASD has a prevalence of 3% in the general population. And previous studies have found that genetic factors could explain between 50 and 80% of the different cases. The problem is ASD is, like I said, a complex disorder with a wide range of genetics risk. And some previous analyses predict that around 1,000 genes could be implicated or associated with this disorder. So the researcher tried to identify these different genes. But because these variants were most of the time really rare, the association of these studies was really difficult, and only a few of them were associated at the level of at the genome-wide level. And then most of what we know on the ASD risk genes, ASD risk comes only on these few de novo variants hitting these genes, and we found that already there is 280 genes that were associated with ASD. And the thing is, to better understand the ASD pathobiology of all this, the ASD pathobiology, the researchers have tried to look if there is some convergence between those different genes on the cellular pathways or cell types. And for example, in this research, they found that the 74 genome-wide significant associated ASD genes were enriched in 4 different prenatal cell types, which are the excitatory, the maturing, excitatory, and inhibitory neurons, and only during the prenatal stage. The problem is, like I said, most of these variants were only studied through de novo analysis. And the problem is rare SNVs, inherited SNVs, and CNVs also have an impact on the ASD risk, but it's too difficult, or it's really difficult to study them because of the statistical power issues, because their effect sizes will be lower than what the de novo variants will have been. So the biological functions behind these genes and their association with ASD remain unknown. So the overall knowledge gap is that the cell type association were mostly performed on the few genome-wide significant genes, with show that there is still a huge part of the pathobiology that remains unknown, and also on the enrichment analysis, only the prenatal stage was studied because it was the cell type the most accessible for these studies. So the idea of my project is to identify all the different cell types and the developmental epochs associated with ASD when their genes are disrupted by any rare variants, by using another method, not relying only on the genome-wide association. So to do so, I used 150,000 nuclei, single nuclei coming from cortices, prefrontal cortices of 30 individuals, aged between the 22nd gestational week and 40 years old. And these 150,000 cells were divided into 18 different cell types and 6 different stages. Then I assigned all the 20,000 protein coding genes to the cell types to define them. So for example, each cell type will be defined by its top 5% most specific genes based on that score. And then I try to redo the same analysis done before, using, for example, here the 600 most specific genes to the L23 CUX2 neurons, and see where the 280 high confidence ASD genes were enriched or not. And for example, like I said, for the L23 CUX2 neurons, excitatory neurons, we have 6 developmental stages going from going from the left to the right, with on the left the prenatal stages and the adult stage on the right. And we can see that, like expected, the HC-ASD genes were enriched in the prenatal time. And when we do all the things for the 91 different developmental cell types, we found that once again, yes, there is an enrichment of HC-ASD genes in the prenatal epochs on the left column, but we've also found a second enrichment at the infancy stage, which is the third one, and this infancy stage is between 1 and 3 years old, and that's also the age of the onset of autism. So that shows that there is also some alteration that happened at this time. But the problem is, once again, we are only studying the 280 genes known for SD, and still there is a lot of potential risk that remains to study. So for that, we developed the functional burden, and the idea is, we assume that for each gene has an effect on ASD that is either positive, null, or negative. And within a certain biological function or cell type, there will be a convergence of this effect. So to compute the cellular burden, for example, in this example, we sum all the different variants altering all the genes assigned to this cell type, and then we compute a logistic regression to get the ASD liability of this group of genes together. And we do that for all the 91 different cell types to have the full picture of the ASD liability. So for this example on the ASD, I used all the rare variants, so with the frequency below 1 for 1,000 in the Spark dataset, which is composed of 141,000 individuals, and 55,000 of whom have ASD diagnosis. And we also divided all the different variants based on their consequences, so as defined by the VEP. And we can see that all our variants will give us enough power to study the majority of the protein coding genome and the genes expressed in the brain. And also, the thing is, now that we have enough power, we can study the genes, or we can study the variants at their variant type level. So for example, here we study only the effect of the deletion on the risk for autism. And once again, so if a cell type is in purple, that means that when their genes are deleted, their effect on they increase the risk for ASD. And for deletion, we found that there is most of the association or the risk of the increased risk is for excitatory neurons and inhibitory neurons also at the prenatal stages, a bit like the previous enrichment studies. But when we study and look at all the 6 different types of variants, we see that all the different patterns are completely different. For example, for duplication of the exact same cell types or gene sets, we found that the signal isn't this time mostly associated to, So this time we can see that it's mostly localized during postnatal stages and mostly in the excitatory neurons. And for the stop gain and frame shift, we see that we have a broad range of effect that is distributed almost across all the different cell types. And I forgot. So the thing is, okay, we have all this cellular liability, but it can still come from all the 280 genes we already know. So what we did is we removed all these 280 genes from our analysis to see if there is any cellular liability that remains. And this is what we found. So for deletion and duplication, the signal remains roughly the same, just we lose a bit of power, and we still have a lot of different cell types that are really interesting to study, because none of this liability is coming from the known ASD genes. And the thing is, if we look at the concordance between the previous liabilities found and the new one, we found that there is a high concordance for deletion, duplication, missense, and splice variants, because these 280 genes were rarely identified with these two types of variants. Only the stop gain and the frame shift were used to define these 280 genes. And even for those, we see that the liability remains, and we have only a 20% drop of the effect sizes, showing that even for those variants, still a large amount of the signal or the liability at the cellular level remains to explain. So to go beyond just showing which cell type are really associated to ASD or not, we decided to focus on microglia. Why? Because this cell type is expected to have an effect on ASD, because it's one of the cell types that is most dysregulated in ASD brains compared to controls, but it was never found associated with ASD genes. And even in our enrichment analysis, it was one of the only rare cell types that was never associated with. And the thing is, when we use the cellular liability method, the functional burden method, we found that there was an association, and even after removing the known ASD genes, the effect remained. So to do that, we developed a bootstrap version of the functional burden, where we downsampled the gene sets, the gene set associated to the microglia, and tried to find which genes are overrepresented in the significant subset of the genes, of the microglial specific gene sets. So we did that, and we found that there was 44 genes that were significantly enriched in ASD, significantly enriched among the significant downsampled subsets. And among these 54 genes, 40 were never associated with ASD before in any study. And we can see also that with the expression matrix in the middle, that the expression was really specific to microglia, which is represented by the gray column here, showing that maybe the effect is really due to their impact on this cell type and not on the others. So to conclude, the FunBurd method can identify the broad cellular liability for complex disorder in the context of rare genomic variants, and by collapsing on the biological function, we provide enough power to study rare variant types individually, and the bootstrap implementation of FunBurd can determine the gene that contribute the most to the cellular liability. And for ASD, we can find that the liability for ASD expands far beyond the prenatal epoch like expected before. And for the microglia, even if before we expected that it was only a consequence of the perturbation, we can now see that the genes specific to this cell type also have an effect on the disorder. I would like to acknowledge both labs I was working with, the Jacquemont and Nowakoski's labs. And if you want to try this this tool. It's available on GitHub, and a paper showing how showing all the different results on ASD will be released really soon and announced on the GitHub. Thank you. If you have any questions.
Time for questions. I'm not sure. You're shy.
Okay. No question?
Yeah.
Lovely talk. I wondered if you'd stratified for the analysis, somehow stratified, because ASD, like you showed, is a broad spectrum, and whether you'd implemented stratification somehow into the model, and you may see, cell type specific signals with certain patient groups, but
Yeah,
I don't know how easy that might be.
So we try to do some stratification either on the sex basis, because we know that there is I didn't mention that, but there is a high differences between male and female for ASD, and the same with ASD with intellectual disability or without. But the thing is, we don't have, even with 1,000 individuals, we don't have 100,000 individuals, we don't have enough power. And when we stratify, we don't see anything significant. So yeah, we tried, but nothing pop out about that.
So a call for more samples if people have.
Yeah.
So, thank you, Thomas, again.